All right, so what we actually have is a small scale dairy processing cooperative that wants to test out how they can actually carry out fermentation of these dairy products. So what they actually do is that they choose two methods, method A and method B. Method A is when they're using traditional culture that slowly ferments milk over 24 hours at ambient temperatures. Okay, remember they're interested in producing yogurt. So yogurt is produced as a result of fermentation, and this fermentation has to happen with the help of enzymes. So they're using um, traditional culture to get that enzyme that can carry out that fermentation. And then that the second method is when they introduce a purified enzyme called lactase to break down lactose. Remember, lactose is the sugar that we find in milk. So after breaking down this lactose, the next thing is that they ferment. The fermentation basically happens by introducing this lactose in a fast-acting bacterial culture. After several cultures, they find out that B produced yogurt within 4 hours. And remember the other first one where they had traditional culture, the milk fermented in 12 hours. Okay, so but some consumers are complaining about the altered taste in method B, altered taste and texture. So the cooperative now needs to make a decision on what basically to use. Is it A or B? So they are now seeking for biological insights to be able to optimize production without compromising quality. Okay, so you can use the traditional culture. It produces good taste but takes long. Okay, and then you can use the enzyme, a purified enzyme, produces the yogurt very fast, 4 hours, you have your product, but the taste and texture is altered. So what do you do? Okay, so the first question is can you describe the mode of action of enzymes in reference to the enzyme lactase? Can you use a diagram to illustrate your explanation? The second question is can you compare the biochemical and environmental factors that affect enzyme activity in method A and B and discuss how these may influence the difference in the yogurt production time? And then can you evaluate the advantages and disadvantages of using purified enzymes versus microbial fermentation culture in food processing? We want you to consider aspects of efficiency, cost, quality, and safety. So this is an excellent question. This question basically introduces us to a number of concepts. The first concept that this question introduces us is the concept to do with the fact that, yes, our bodies or organisms actually have enzymes, and these enzymes are biological molecules that speed up the rate of chemical reactions. These enzymes are usually proteinous in nature, but these enzymes can actually be manufactured or can be synthetically made. And that's what we are talking about when we refer to purified enzymes. Okay, so these enzymes can be made and can be utilized in industrial activities such as yogurt production that we are seeing in this case. Okay, so of course, because these are synthetic enzymes, we need to make sure that they actually act in when the conditions are optimal. So those conditions are which ones? Conditions of temperature are very important. Like in this case, we are talking about ambient temperature. Okay, and then this ambient temperature is influencing the rate of this enzymatic reaction, eventually influencing the rate at which you have the final product, which is yogurt. Okay, so those are the insights that this question actually brings to us. So let us first look at the slides and then come back to this scenario. So enzymes are basically proteinous molecules that act as biological catalysts. That means that they actually increase the rate of biological reactions or chemical reactions in the body. All organisms have these enzymes, okay, because you have a number of biological processes that take place in the bodies of these living organisms, and these need to be occurring at a faster rate. So the enzymes actually do that. So we can say that the enzymes are biological catalysts. So these enzymes speed up the rate of chemical reactions in the body. If these enzymes are not available, it could mean that the chemical reactions in the body could actually be occurring at a very slow rate. Okay, so without these enzymes, you could actually have problems, because we know of genetic conditions that are associated with the lack of enzymes. For example, a condition known as lactose intolerance is a condition associated with the lack of lactase. So these people cannot actually digest lactulose. So we say they have lactose intolerance. So this is one example to illustrate how enzymes are, because if some enzymes are not there, then you're going to have problems as a human. Okay, so enzymes are very, very many. They catalyze a number of reactions, and we can categorize them according to where we find them, what they do, and then also can categorize them according to their mechanism of action. So according to where we find them, we can say we have extracellular enzymes. These ones are found outside the cells. So usually they are secreted by cells and then act outside the cells. And the digestive enzymes actually are extracellular. And then we have those that act within the cell. So we say that they are intracellular. Okay, intracellular enzymes. So all the enzymes of, for example, respiration, these are enzymes that act within the cell. Okay, whether cytoplasm or within the nucleus. Okay, and then when we see a name of an enzyme, it should depict to us what that enzyme actually does. Okay, so for example, all enzymes always have that suffix. The word at the end implies that they are enzymes. So if you say amylase, you can say that the last 3 letters are ASE, that is the suffix. Okay, so lactase, you can see that ASE. Okay, and many other enzymes. So when you see such, you should be able to know that we are dealing with an enzyme in this case. And like you have seen, lactase, that is an enzyme acting on lactose. Okay, lactose is a sugar that we actually find in milk, and in our scenario, it's actually what we are talking about. Okay, so usually, if you look at the name of the enzyme, it tells us that the suffix is that it's an enzyme, ASE, and then the name may suggest which substrate it actually acts on. Okay, so for example, you can have an enzyme called ATP synthetase. ATP synthetase. So that means that it is an enzyme that help in synthesizing ATP. Okay, and also, for example, when we say lactase, lactose. and then the enzyme part. Okay, so that's what we can know, okay, from the names of these enzymes. So we can categorize these enzymes based on what they actually do. So when you look at an enzyme, its name will always suggest what it does. Like the example I've just given, ATP synthetase. So you just know that this enzyme is very important for synthesis of ATP. So you can have oxidoreductases. That means that these enzymes are responsible for redox reactions in the body, because oxido, that is oxidation, reductases, that is oxidation and reduction. And then we have groups of enzymes that we term as transferases. They transfer groups of atoms from one molecule to another molecule. And then we have hydrolysis. This one, they carry out hydrolysis. And hydrolysis in our case is when water is used to split a molecule into its simpler components. And then lysis, which is basically breaking down a molecule. Okay, that is a lyase enzyme. Okay, so they break down and such that you have other molecules being released. And then isomerases, they cause what you call isomerism. So with isomerism, it's basically a rearrangement of the atoms on the same molecule and basically moving them to other parts. Okay, so that is what happens. And then ligases, to ligate means to join, ligate, okay, means to join. So ligases do join molecules together. Usually energy is actually utilized. So these enzymes have a number of properties. Remember, we said that they are protein molecules. And to be more specific, we can say they are globular proteins. Okay, so that means that some of the properties of proteins that we have actually looked at before actually affect these enzymes. So when talking about proteins, we talked about denaturation, which can be caused by acids and then also high temperatures. Okay, so it's the same thing that we have with enzymes, that they can actually be affected by high temperatures and also changes in the pH. Okay, so not all enzymes actually have the same efficiency. If I talk about efficiency, this is what we are meaning by turnover. So what does this mean? That, okay, the rate at which you get products out of the enzyme substrate complex, the rate at which the enzyme works is what we are terming as efficiency. So we have some enzymes that are very, very fast at working, so they have high turnover. And then we have those that are quite slow. So that's what we are meaning by saying that enzymes have varying turnover numbers. Okay. Enzymes are not destroyed by the reactions that they actually catalyze. So that means that they actually remain unchanged, just like what happens with inorganic catalysts. Okay, so that means that they are released at the end of the reaction. So what do they usually do? Is that enzymes actually lower what we term as the activation energy, the energy that is required for reaction to basically start. And this activation can be reversible or irreversible. Okay, so we have enzymes that can catalyze forward reactions, getting the products, or you have catalysts or enzymes that are actually organic catalysts in this case, or enzymes that catalyze backward reaction, that from the substrate, from the products, you want to get the substrate back. Okay, so we can have those reversible reactions, and you have enzymes both promoting forward, and then you have another enzyme promoting the backward reaction. Enzymes actually act on specific substrates. Okay, the meaning of that is that they don't act on every substrate. And that is why actually the name suggests that, for example, lactase, that means that acts on lactose and you don't have any other substrate that it will act on. And we'll see why that basically happens. Okay, like I said earlier on, enzymatic activity is influenced by temperature, but also by pH. And when we talk about the influence of temperature on enzymatic activity, we always need to talk about, for example, what we term as optimal temperature. Optimal temperature is the temperature at which the enzyme is working efficiently well, okay, very efficiently. And then suboptimal temperature is when it is not really working properly. So how does temperature influence enzymatic activity? At low temperature, you don't have the enzymes and the substrates colliding at higher rates to cause the formation of enzyme substrate complex. Okay, but when you increase the temperature, the rate of reaction increases because the rate at which you are forming enzyme substrate complex is very high, and then that results into an increase in the rate of reaction. But at optimal temperature, that is when you have maximum collusion, and that's when you have the enzyme basically working efficiently. Beyond that, you will have the enzyme changing its structure because of denaturation. And that means that after the optimal temperature, you have a dramatic reduction in the rate of reaction. Okay, and that is what you see in on the graphs. You will see that you have two enzymes, but both enzymes are actually having different optimal temperatures. Okay, but both enzymes that we see in the first graphs, these enzymes drop their rate of action drops dramatically after the optimal temperature. Okay, so the pH, also you have enzymes acting at the optimal pHs. These pH ranges can actually be different for different enzymes. We have those that prefer alkalinic media, and then we have those that prefer acidic media. Okay, so we have enzymes that prefer close to neutral pH. Okay, so like what we see on our graph again, you say that pepsin works best at around pH of 2, that is very, very acidic, and then carbonic anhydrase works optimally around pH of 7, which is basically neutral, and then we have chymotrypsin working best at around the pH of 9. So this is again the fact that you have different enzymes working at different pH levels. What causes this? Remember, these enzymes are actually proteins, and these proteins can have charged parts. Okay, and when you have charged parts or charged groups of atoms, it is likely that when put in a particular media, you will have interaction of ions with those parts, and that is what causes denaturation. So with denaturation, the shape of the enzyme is altered, okay? And when it is altered, the active site will no longer be able to fit the substrate, and that's when you don't have a reaction basically happening. And it's the same thing that happens with the pH or with the changes in temperature. At beyond the optimal temperature, you will have denaturation. With denaturation, still you will have change in shape of the enzyme, and then the active site will be altered and the substrate will no longer be able to fit in. Okay, so as a matter of highlight about how enzymes work in relation to the scenario that we have, is that the enzymes will have to form a complex between the enzyme and the substrate. okay? And then you'll have reactions happening. That's what you would expect, and that is how temperature influences. So going back to our scenario. Okay, so coming back to our scenario, you are basically having part A, which is asking the mode of action of enzymes. At least in this presentation, we know that enzymes have to form a complex with the substrate, okay? And that means that you need to have an active site on the enzyme that the substrate will fit in. After the substrate fitting in, and then the bonds of the substrate being broken, and then you will have the products being released. At least that is what we can talk about now here. But of course, in our subsequent presentation, we shall look at the lock and key model of enzyme activity, and then also induced fit mechanism for action of enzymes. We shall look at that shortly. Okay. And then part B, about the biochemical and environmental factors that influence enzymatic activity in A and B, and show how this influence. And in this case, the factors that will influence the enzymatic action, in this case, we are having an issue to do with the temperature, okay, and then we have an issue to do with the media. How does the media come in? The media here, in case A, we are having traditional culture. That is the medium through which you have the enzymes actually working. In the second case, we have fast-acting bacterial culture. So these cultures actually will influence the rate of action of these enzymes, because these media actually may not be of the same pH, because remember that pH actually influences the rate at which enzymatic reactions actually happen. Okay, so that is the point that you have to consider. But the second point is basically the temperature that we can relate to easily here with this scenario. Okay, so those are basically biochemical factors and also environmental factors. Advantages of using purified enzymes. At least for this case, it's like you know how much enzyme you need to add. That gives you control over the rate at which you're going to have the fermentation happening. Okay, so you have good control over quantity of the enzyme used. But for method A, you don't have that control. That is an advantage and a disadvantage on the other hand. And then when you use this synthetic purified enzyme for the method B, it changes the taste and texture. Okay, yes, the thing will happen very fast, the fermentation will happen very fast, but you have that disadvantage of changing the quality and the texture of the final product. So you have issues there. Generally, if we use the traditional culture, what you get is actually safe for use. But if you use this purified enzyme, you may need to take into consideration other measures to basically assess quality, such that you know that you have not gone outside what is expected. Okay, so thank you.